to ask some devastating questions, and that have been suggested to me and others I've thought up. And I want to go with almost with the theme that Marla ended up with, but also that Steve had throughout his presentation, is this construct observation of variability, heterogeneity, diversity in the patient population. And at times you think that's going to end up being chaos, and yet at the same time, I wonder, and I got some suggestion from Steve's presentation as well as from Marla's, is there in the heterogeneity, in that diversity, in the variability in the presentation, is there a diamond? Is there something there that's important? Steve, and I'm going to turn to you on that.
Yes, but not always. That's what we do as scientists. We look for outliers to kind of guide the way forward and develop hypotheses. So I can think of a few examples, which I did not show. Actually, I took one of the slides out just before my presentation. I mentioned the one region that's a copy number variant that's been defined for many years. It's a region on human chromosome 7. I studied this in my PhD thesis. And if you're missing one copy of this 1.6 million chemical base region on chromosome 7, it encompasses 16 genes. You have a disorder called Williams syndrome, Williams-Beuren syndrome. And it's very, very predictable of what the outcome is, including the behavioral outcome. And then more recently, it's been found that if you have 3 copies of that gene, you seem to have a neurodevelopmental condition that is very reminiscent of autism, and that what some people are now actually calling autism. So too much leads you in one trajectory, and too little leads you in a different trajectory. These are pretty predictable. So in a way, I do think, I agree with what Marla said, but I think we have to remember that our DNA is a blueprint, but we're very hardwired. That's why you look like your father, and you look like your mother. That's controlled by the genes. But it's true. We in the pursuit of scientific investigation, we follow these types of leads. And I gave you one example. There's lots of others. We have some unpublished data now where we're getting a little bit more specificity. I was talking about it today in my presentation at the meeting. We're getting more specificity as we increase the resolution, and we have these new technologies that get us down to the specific genetic alteration, not just kind of an assumption of where it might be. And if we have good clinical details, we can make much more positive correlations. So I think there are more examples coming. But it's not a perfect science. I mean, we're studying behavior here with all of its complexities.
Marla, did you want to add anything to that based on your comments, or are you okay with that? And I will go to another question.
I would say that for behavior, I wouldn't want to say that we're hardwired as much as maybe some physical characteristics. Sorry. I would say that the term of hardwired in a DNA blueprint, one has to think about which particular traits you're talking about. And so for something like a physical characteristic, having a hitchhiker's thumb or not, I don't. It's a single gene trait, or some traits that are single gene that we know about, like Huntington's disease or cystic fibrosis, then we can talk about a genetic blueprint. But when we're talking about traits like normal variation in behavior, and maybe some of the behavioral traits for some of these disorders, it isn't a one-to-one mapping necessarily of gene and behavior. We have mutations in an autism gene, and yet it's not completely penetrant, right? That child doesn't end up with it. So the genetic gives us a predisposition, but I think we don't want to give up on understanding things about the environment that could be interacting somehow with that predisposition.
That's true.
That would be
that's what I said.
I'm just leaning.
I mean, we know through behavioral studies in animals and in humans that we are hardwired, or maybe a better term is we're soft-wired. But in fact, a lot of things, and each of us can tell stories about our spouses, but my spouse would say, "You're stubborn, you got it from your dad." And I have many attributes that are probably hardwired based on my genetics.
So you couldn't change your stubbornness?
No, I can't. I have no control.
And this is how science advances. We've had 2 days of this. A little more sophisticated than "No, you're not." No, that one doesn't. That argument is well, it does come out every now and then in science. So I want to go to Evdokia. Evdokia, you're a clinician, yet you're leading the PAWN network. And how is basic science going to assist you or assist clinical work in general? Why basic science?
Well, I'm a neurologist, and when I was training my supervising neurologist, every time I saw a child, he would say, "Where is the lesion?" So we got trained to identify the lesion. You know, if a kid had a stroke, you looked for the signs of the stroke, and then you figured out where in the brain that stroke localized. When I saw a child with autism spectrum disorder, my supervisor asked me, "Where is the lesion?" And I had no idea where the lesion was, and very quickly realized that without understanding where the lesion is, there is very little opportunity for innovative therapeutic development, for making big improvements in the way we treat kids, because we don't understand the biology. We don't understand the lesion. So what basic science does for me is it gives me a lesion. Once we have the lesion, once we know what the molecular target is, the biological target is, then we have tools, and there is huge technology development happening at the same time that allows us to get much better at developing new tools to actually develop treatments that target that lesion. But we need the lesion.
Right, so lesion as a metaphor.
As a metaphor
for understanding the mechanism.
Absolutely.
What's underneath,
Yes.
which is the issue. And I think if you think a bit about the debate that Steve and Marla was happening, was exactly that, I think, back and forth, understanding the mechanism, the hardwire, with what's happening on how you interface with how this is expressed. And I think that may be another way of phrasing that. Marla, I want you to think big. Where are the breakthroughs going to come through? How do we get them? Where are we going to go?
So I think one of the things that has to be done, and I know it's being started already, is to bring people from wildly different fields and areas together who wouldn't normally find themselves in the same room at any meeting, to work at the edges of their discipline. It sounds like a CIFAR model, but to really push them to address the questions. And that way, and it's the same in my lab, you get out of your rut of thinking in a certain way, and you hope to make big innovative discoveries. So I would say that's how to do it, and there are many groups starting to do that, including here in Toronto at Sick Kids.
Right. In other words, thinking differently about approaches, and sometimes we need to have the conflict to be able to say which is one or the other, and why essentially why our workshop for 2 days was to bring in people of diverse backgrounds. Steve, I'm going to go back to you, and I am going to end up then, Evdokia, with a devastating question for Evdokia that we're all dying to ask, but so I'm letting her prepare herself there. But Steve, I want to go back to the thing that you said, because I'm a neuropsychologist. You described the whole issue of behavior, and you talked about the diversity from the social cognition versus the, quote, executive. And in my domain, that would be, both of those are related to frontal lobe functioning. This is the area of research I do. And yet, we've also taken a look at, and we've heard over the 2 days, this whole issue of networks and systems. So talk to me, I mean, even though it's not your field, so how do you see that area pursuing, or do you feel that that's not a fair question to ask you?
That's definitely not a fair question to ask.
I told you I was going to ask.
Why don't you talk? This is your area of expertise. Maybe what I will say is, you know, I presented a presentation of the things that really excite me, and I think we've seen more progress in the last few years that have really revolutionized the way we think about autism. Okay, I think the concept of molecules meeting at the synapse and the development of animal models based on human mutations that now seem to be reversed in phenotype using specific drugs or training behaviors and things. I think this is really exciting. It's what we've been looking for. But in saying all of that, we've been looking under the lamppost. We have these massive data sets that we can generate by genomics, proteomics, imaging, and we tend to look for things that we find simple or can explain. So maybe I'm being a politician here. I'm changing your question to come back to the question you asked Marla. Really, and this is what I think the reason for the meeting was, is to bring mathematicians and statisticians and modeling people to the fore so that we can actually really start to understand the depth of the data and what it should be telling us, as opposed to looking for what we think it should be telling us. So that's where we need. That was my last slide. I think that's where the breakthroughs are going to come, at the interface of these disciplines, and thinking about new ideas. So for example, in autism, why are we studying males so much? I mean, 4-to-1 male, we do a collection. We should really be studying females, because perhaps the clue, that Shank-1 family I talked about, is trying to find the genes that are protective, right? That'll give us the clue of actually developing treatments. Maybe easier to protect than actually replace or correct. So those types of bright ideas coming from bright people, but I think using different approaches and exploiting the massive data sets that we can develop technically, but actually bringing them to completion.
No, and that actually is a kind of an answer that I wanted, and I just, as an aside, and it was an unfair question I was asking Steve, but one of the constructs is about thinking broadly, is that some of the work that I've done, I've actually borrowed from the autism research to do in adults with stroke. So this idea of pushing the boundaries across, and you can borrow from anywhere to come across with ideas that help you to understand the question that you're pursuing and the patient population that you're pursuing. So I think you're spot on with your response. Now I'm going to ask Evdokia the question that everybody wants me to ask, that everybody's dying, will there ever be a cure?
So, I mean, it's a theoretical possibility, but I don't think that should be the focus, for a couple of reasons. One is our brains, the brains of kids who have autism spectrum disorders, get wired differently from the beginning. So by the time a baby is born, their brain is wired differently. So it's extremely unlikely that we're going to have manipulations that we do after birth that would reverse the original wiring. That's one way to look at it. The second way to look at it is you may not want to change it. So we're all wired a little bit differently. It produces variability, it produces innovation, produces some conflict. Then when it gets generated, good things happen. So we don't really want to be all the same. We don't want to get rid of all the differences. What we want to do is we want to decrease the stress, we want to decrease dysfunction, we want to improve quality of life. So the purpose of our treatment strategies should be to allow the difference, but take away the distress. So it is not like a person who is not particularly socially motivated will become a social butterfly after the treatment. It's just that we want the person who's not socially motivated and is feeling, and is really becoming a little bit of an outcast, to have the ability to interact, to have the ability to have an interview, to have the ability to get a job that actually matches their skills, to have the ability to get satisfaction out of their life. So I think the goal should be focusing on quality of life and removal of distress, and less on whether in one day it would be theoretically possible to cure the disease.
Thank you. Steve, did you want to?
Well, I don't disagree with anything that she said, but there are parents who want their child treated towards a cure. Certainly severely autistic conditions that are medical genetic disorders that can lead to a very debilitating lifestyle. So I don't think we should forget that also. In fact, that really drives our research forward. We want to understand all the things I talked about, but ultimately we want to provide better treatment options for these people who really need to have medical help.
On the entire spectrum, from what you've said to what...
This is the absolute challenge in the field.
Right.
I mean, we're dealing with, as I showed on my slide, a disorder that ranges from a Glenn Gould to a severe medical genetic disorder.